All right, so now we're going to go through paper 4. This is the extended theory option for biology. It's IGCSE from Cambridge International Examination, CIE, May-June 2018, and variant 2, so this is 0610/42. Let's get started.
Are you ready to begin?
Question 1A. Red pandas, Ailurus fulgens, and humans have a similar arrangement of teeth. Figure 1.1 shows a section through one tooth of a red panda. Figure 1.2 shows the side view of the lower jaw of a red panda. So we have figure 1.1 with a tooth and figure 1.2 with a jaw. 1. State the names of the structures labeled A to F in figures 1.1 and figure 1.2. All right, so first of all, we have this level here. That is dentine. Then we have the stuff that holds in the tooth all the way around the outside, and that is called cement. And we have the teeth at the front of the jaw. These are known as incisors. Then we have this big pointed tooth, that's a canine. And we have these ones at the front of the back section. These are premolars. And these ones at the very back are the molars. Okay. So 1 was a dentine, or A was a dentine. B was a cement. C were the incisors. D were the canines. Well, 1 canine. And E was premolars. And F molars. Sorry, there's only actually 1 canine being shown in that picture, so probably it should be singular. That's the end of this part. 2. State the type of digestion that breaks down large pieces of food. Well, we have 2 types of digestion. One is chemical, one is mechanical. Chemical digestion breaks down small pieces into their molecules that can be assimilated. And so this is mechanical digestion. Mechanical digestion. B. Food that sticks to the teeth can be used by bacteria for anaerobic respiration. This type of respiration releases a substance that can cause tooth decay. 1. State the type of substance released by the bacteria during respiration that causes tooth decay. And the type of substance, not the actual substance, is an acid. 2. State the names of the 2 parts of a tooth that are dissolved by the substance released by bacterial respiration. The 2 types of the teeth are the enamel and the dentine. So this is the enamel layer. The white layer is the enamel. And the gray layer here is a dentine. And if you get down, if the cavity gets all the way down to here, then those are the nerves, and that's very painful. C. The teeth of red pandas do not decay as much as human teeth. Suggest the component of a human diet that causes teeth to decay as a result of bacterial respiration. Well, that would sadly be sugar. Humans eat a lot of sugar, and bacteria really like the sugar. Okay, pandas don't eat very much sugar. 2. Mangrove trees are hydrophytes because they grow in water. Figure 2.1 shows a young mangrove tree. And if you see, you have your tree, then you have all these really interesting little roots coming out here. A. An adaptive feature is a feature that increases the fitness of an organism. Define the term fitness. Okay, when they want you to define the term fitness, they're not talking about physical activity, because obviously plants don't do physical activity. Okay, they're talking about survival fitness. So when you're talking about defining the term fitness for adaptation, you're talking about the probability or the chance that an organism will survive and reproduce in the place where it lives. The key there is it needs to reproduce, because if it survives, gets to an old age and dies, then that's not actually as fit an organism as a plant or animal that survives for a short bit but reproduces so that the species can continue in that time. So fitness is the probability that an organism will survive and reproduce in its environment. 2. Mangrove trees have many aerial roots and floating seeds. Suggest how these adaptive features allow mangrove trees to survive in water. So the many aerial roots, those are these structures here that come out from the plant. They have quite a lot of them. Those are the aerial roots. So basically, they're roots that grow in the air. They don't grow under the ground for the most part. Okay, and what do? They have a few purposes. Okay, one of them is that if the water is moving, then it allows the plant to be anchored. So this is it makes the base very wide, so it helps the plant get anchored in flowing water. Other things they can do is if the roots are in the air, then it allows for the root cells to be able to do aerobic respiration and gas exchange so that they can absorb some oxygen. Okay. For the floating seeds, you might notice this plant is surrounded by water. So there's really only two ways these seeds can easily get dispersed. They can be blown away on the wind, but the plant is not very tall, or they can float away. And floating away looks like the easiest option. So the aerial roots, big thing is to anchor the plants in flowing water. Okay, it says many aerial roots, and that helps with that. So as we said as well, they can also be used for aerobic respiration and for gas exchange. They can absorb oxygen. For floating seeds, it allows the seeds to be dispersed far away from the plant. So it allows the seeds to be dispersed far away from the plant. And the purpose of this is it avoids the competition with the parent plant. So they can germinate and grow without having to compete for all the nutrients and sunshine and all that with the parent plant. Another thing another benefit of floating seeds is that the seeds can get oxygen from the air, and they can use this oxygen to germinate. But only put down one answer for each. It's a 2-mark question. They don't want you to put down 2 answers for each. B, figure 2.2 shows a food chain in a mangrove forest. So we have a mangrove tree, and then a fiddler crab, and then a seagull. Remember, the arrows point in the direction of energy transfer, so that means the fiddler crab eats a mango tree, the seagull eats a fiddler crab. So table 2.1 gives the number of organisms and their biomass in a mangrove forest. So in mangrove trees, it might be 1,000 mangrove trees, but there might be 7,500,000 fiddler crabs and only 150,000 seagulls, which is a lot more than the trees, but less than the fiddler crabs. And then it also gives a biomass. So mangrove trees have a much larger biomass than the fiddler crabs. If you put all these organisms together, dry them out, then they will have a much smaller mass than the mangrove trees. The seagulls will have a much smaller mass than the fiddler crabs. 1. Estimate the biomass of one fiddler crab in grams. Okay, keyword there, in grams. And if you notice this is biomass of organisms in kilograms. So you have to be able to convert that. Make sure you read these fine details. Don't just skip past it. So, and this is another line that is easy to miss. Write your answer to 2 significant figures and show you're working. Okay, so the biomass of one crab equals the biomass of all of the crabs put together divided by the number of crabs. So the biomass of one crab equals the biomass of all crabs divided by the total number of crabs. And as we said, that's in they want it in grams and it's in kilograms, so we have to multiply that by 1,000 because there's 1,000 grams in 1 kilogram. So we have 8,000 kilograms divided by 7,500,000 organisms times 1,000 grams per kilogram. Okay, we cross out the kilograms. So that equals 1.06666666. Oh, so 1.07 grams per organism. Okay, we don't need to write in per organism. They have it in grams because they're asking about how much is one fiddler crab weigh. Okay, and the key thing here as well, it said to do your answer to two significant figures, so that equals 1.1 grams. There we go. 2. Sketch a pyramid of numbers using the information in table 2.1 for the chain shown in figure 2.2. Write the number of each trophic level in the appropriate part of your pyramid. All right, pyramid of numbers. So if we look at this, we have 1,000 organisms. That's small. That's the smallest. And we have 7,500,000 fiddler crabs, largest, and 150,000 seagulls, middle. Obviously a pyramid of numbers, pyramid of biomass, they're just rough estimations of how much they are unless you're given a graph to do it on, the graph paper to do it on. Okay, so we make a small bar, then a huge bar, then a bar in between. All right, and we always put the producers on the bottom of the pyramid. Okay, small bar, then a big bar. You probably want to make sure these are nicely touched, as square as you can get, or rectangular as you can get. Okay, you want to make sure they're approximately the same height. They don't have to be perfectly the same height. And then you want to make a middle bar. All right, that's it. Well, maybe the top one should be a bit bigger. Okay, this is approximately it. It doesn't have to be perfect. You want them to line up about in the middle, be approximately halfway. But this is the pyramid of numbers, pyramid of biomass, unless you're given graph paper to do it on, is a sketch. And the second part of this section, write the number of each trophic level in the appropriate part of the pyramid. So they're saying that the lowest, the producers, would be a 1. Then the primary consumers would be a 2, and secondary consumers would be a 3. All right, so that's all you have to do for this question. You can even use a ruler to make it nicer. I don't believe they take marks off for it, though, if you don't use a ruler, but it's nicer to use a ruler. And you don't need to do this one in pencil. It's a sketch. See, keyword there is sketch, and sketch means that it doesn't have to be a perfect biological drawing in any way. 3. Explain why the shape of a pyramid of biomass for the information given in table 2.1 would be different from the shape of your pyramid of numbers. All right, let's look at that table again. So we said before the mangrove trees were the smallest. Now, if we look at the mangrove trees for the biomass, they are the largest. The fiddler crabs are the middle, and the seagulls would be the smallest. That means that the pyramid of biomass would look like, the sketch for the pyramid of biomass would be something like that. And that would be much more pyramid shape. So a pyramid of biomass is always pyramid shaped. A pyramid of numbers is often not pyramid shaped, just like we drew below. Now the question is basically saying, explain why. Why is it pyramid shaped? So first of all, you can say that the pyramid of biomass is a pyramid, while the pyramid of numbers is not. So the pyramid of biomass is pyramid shaped, whereas the pyramid of numbers is not. And there's a few reasons for this. One, you could say that the mangrove tree has a larger biomass than the crabs so that they can provide more food for lots and lots of crabs. So the mangrove tree has a larger biomass than crabs, so it can provide food for many crabs. You wouldn't have one crab only feeding off of one mangrove tree. You'd have many crabs feeding off of one mangrove tree. So the mangrove tree has to be a lot larger than the many crabs. Another thing you could say is that the seagulls have a larger biomass than one crab, so each seagull needs to eat many crabs. So you can say each seagull has a larger biomass than one crab, so each seagull needs to eat many crabs. And that explains why the seagulls have a smaller bar for the pyramid of numbers than the crabs do. And it explains why the mangrove trees has a larger bar on the pyramid of numbers than the crabs do. So those two statements explain why the pyramid of numbers is the way it is. Now we're saying about the pyramid of biomass. You could say that the total crab biomass, the dry weight of all the crabs basically, is larger than the total seagull biomass. So the total crab biomass is greater than the total seagull biomass. So, and the final point you can put down, I know that there's 4 here already. If you missed one of these other points, that's okay. This has an explain why it is what it is for the information given in a particular table. Whenever it really says information given in a table or a graph, what it wants you to do is quote some numbers. It likes some comparative data. So it likes you to know that the examiner likes to know that you can actually read the table properly. And whenever you quote data, you have to put in units. So an example of a data quote is, to support one of these things you've already said, you could say that the total crab biomass is 8,000 kilograms, or the total seagull biomass is only 1,200 kilograms. So the total crab biomass is 8,000 kilograms, whereas the total seagull biomass is 1,200 kilograms. As long as you've quoted some data that supports one of these other statements, then you should be okay. It doesn't have to be that exact one. All right, and whenever you quote data, you always have to include the units. It's useless without the units. So whenever it says explain using a table or a graph, put in a data quote, and that is generally a free mark. Question 3. Aphids are insects that feed on the phloem sap in plants. Figure 3.1 shows a diagram of an aphid with its mouth parts inserted into the stem of a plant. Okay, so this is an aphid, a type of insect, and its mouth parts are syringe-like, and they go all the way down into, this is the phloem of the plant, and as it says here, this is the xylem. You can even see that it's a xylem because it looks like it has the lignin around the vessels. Okay. A. The mouth parts of the aphid reach the phloem tissue of the stem. 1. State the name of the foods the aphids could suck out of the phloem tissue. Okay, so what they could suck out of the phloem tissue is basically what's in the sap, and the sap contains things like sugars and amino acids. So the type of sugar that's transported in plants is sucrose. All right, and the type, and as I said, it also transports amino acids that will become proteins. Okay, so sucrose and amino acids. 2. Explain the role of phloem in plant transport. Use the words source and sink in your answer. So explain. So phloem is responsible for translocation. So phloem does translocation of sap. So what is translocation? Well, basically it moves the sap from place to place where it's made to where it's needed, the source to the sink. So it does that because it allows the sap that has the sugars and amino acids in to move in both directions. Obviously xylem only moves in one direction. So it allows sap containing sucrose and amino acids to move in both directions. You don't really have to repeat that it contains sucrose and amino acids. You kind of answered that in the part just before that. So you can say it allows sap to move in both directions. So that you can use a different term. You could say it allows for bidirectional movement. You want to sound a little bit more scientific about it, I guess you could say. So rather than saying moving in both directions, you could say bidirectional movement, but both are okay. So sap is moved from the source where it is made or taken from storage to the sink, which is where it's needed. So sap is moved from the source where it is produced, so here's a keyword, source, or taken from storage, so it can be either made, say in the leaves, or taken from storage, being in, say, a tuber, a root of a plant, to the sink, so you've used the word source and sink, and the sink is a region of respiration, storage, or growth. So all those are important. And you can give an example of a source and an example of a sink. So an example of a source is a leaf, and an example of a sink is new growth. But obviously, these examples only apply in the summer, when you have leaves and new growth. At other times of the year, say the spring, when you don't really have any leaves, the source will be, say, a root or a tuber, and the sink will be the new growth. So these things can change. So some organs can be a source and a sink depending on the time of year. So say a root or a tuber can be the sink in the fall. So it's trying to store extra energy in the fall. And in the springtime, when there's no leaves on the plant, it is now the source, because that's where all the sugars to make new growth are from. And you might notice here I put down 1, 2, 3, 4, 5 points for 4 marks. Okay, it's okay to put down 1 or maybe, depending on the question, maybe 2 points extra compared to what's being stated, just in case one of your points doesn't count. But don't go on and on and on. I would always recommend that you write in bullet point so that the examiner, when they're marking their hundreds and hundreds of papers, can easily find your marks. You don't want them to hunt for your marks. If you do need to continue on to another page, don't just draw an asterisk or maybe an arrow. Say, continued on page 3, or whatever the page is, whatever the blank page is. And you can draw an asterisk or arrow if you want after that, but make sure you write the page number. You make it easy for the examiner to find your marks. Although you shouldn't have to write. If you're writing an essay, you're probably writing too much. B. Figure 3.1 shows some of the features of xylem. Describe how xylem is adapted for its functions. Okay, there's 2 points here. We have adaptations and functions. So basically we want to say the functions and the adaptations for it. We want to link the two together. So we can first of all say that xylem transports water and mineral ions. So xylem transports water and mineral ions using transpiration as a driving force. It's not the only driving force, but it's a big factor in what moves the water through the plant. So how does it do this? It's adapted to move water through the plant in many ways. One of them is that it has long vessels that reduce the resistance to water flow. So if the water had to go through a bunch of cells and find paths in between all these little cells, there'd be a lot of resistance in between. But it's a lot easier for it just to go straight up a tube. So it does this by having long, hollow vessels to reduce resistance to water flow. So that it doesn't have any organelles. There's no cytoplasm, no nucleus. None of that get in the way of the water. Another way that it allows the water to flow very easily is that it doesn't have cell walls at the end of the xylem. It just has end plates. So xylem have no end walls between the cells, only end plates that allow the water to flow easily. Okay. So another adaptation you can talk about with from xylem is the lignin. And lignin does a few things. So lignin prevents the vessels, the xylem vessels, from collapsing inward with all the pressure of the water coming up. So xylem has lignin in the walls to prevent the inward collapse of the xylem vessels. That's very important. Another thing that the lignin does is it's in spirals, so it allows the plants to have flexible stems. So lignin is in spirals to allow the flexibility of stems. Another thing you can talk about is that the xylem cell walls are actually waterproof, and they have pits on the side so that the water can flow between the vessels basically to where it needs to go, not just escape wherever it can. Okay, so xylem cell walls are waterproof with pits that allow water to flow between vessels. You could also talk about secondary thickening in the cell walls, allowing the xylem to act as structural support for the plant. All right, but here we have 6 points, and the key with this question is you're linking adaptations with functions. So you're trying to link, when you're trying to say that there's a function and it's how it's adapted for that function. Okay, good. C. Some farmers spray their crops with insecticides to kill pests such as aphids. Explain the benefit of killing pests. Well, there's quite a few benefits to killing pests. Obviously, you don't necessarily want insecticides on your food that you eat. However, there's a lot of benefits to using them. Well, one of them is that killing pests reduces damage to the crops. That's a key bit, and reduces in a few ways, and we can mention those in a second. So killing pests reduces damage to crops. And when damage is reduced to crops, the farmers get a higher yield, and so more money. So this increases the yield of the crops and creates more profits for the farmers. Well, how does it do this? Well, pests eat the sugars and amino acids that the plant needs to grow. So pests eat the sugars or consume the sugars and amino acids that the plants need to grow. So the plants obviously have to produce more sugars and amino acids than they would if they didn't have the pests. And another thing that pests do, which can be very devastating to crops, is that they transmit diseases. Just like a mosquito can transmit some diseases like malaria to humans, aphids and other pests can transmit things like viruses to plants. So this is pests also transmit diseases to plants. Okay, so we have four points here. It's only a 2-mark question. You don't need as much as that, but there should be enough there that you could fill in a full amount to answer it with. Question 4. One of the roles of the Centers for Disease Control and Prevention, CDC, in Atlanta, U.S. is to try to reduce the number of people who are infected with pathogens. The CDC conducted a survey. They asked women which, if any, contraceptive methods they used. A. Suggest why the CDC collected data on contraceptive methods. Okay, there's quite a few reasons they could have used. They could have collected it. They could be collecting it because first of all, there's many diseases that are spread through sexual contact. There are many diseases spread through sexual contact, and you can give an example, and that's another mark. And an example, you can say such as HIV and AIDS, or any other disease that you know is spread by sexual contact. So such as HIV and AIDS. Okay, obviously that's not the only way it's spread, but that is one way. So some of the STIs, or sexually transmitted infections, can be prevented by certain types of contraceptive methods. Again, you can put in an example here. So some of the STIs can be prevented by some contraceptive methods, such as condoms. Don't just say that they can be prevented, because there's some contraceptive methods, such as the pill, which will do nothing to prevent STIs. It will just prevent pregnancy. All right, so remember, contraceptive means it's blocking pregnancy. It's not about preventing sexually transmitted infections. That's just an added bonus. Okay, so the CDC can use the information that they get from these studies for a few different things, such as creating useful education and preventative strategies. And they can also use it to assess the effectiveness of different strategies that are actually being used. So the CDC can use the information they collect to create useful education and preventative strategies, and they can assess the effectiveness of different contraceptive strategies that are already being used to prevent diseases. Okay, that's a long sentence. You could probably divide that into 2 easily. So that is probably 4 marks for 3, and that's okay. So figure 4.1 shows the results of the survey. Okay, so in the first bar, we have any percentage of women in the survey that have used any method, and that is about 78%. The ones that have used condoms, that's about 68%. Contraceptive pills, that's about 16%. And other chemical methods, that is about 6%. Okay. B1, state 2 hormones that are used in contraceptive pills, and so those hormones are estrogen and progesterone. So estrogen and the progesterone. Don't forget that sneaky little O in front of estrogen. Just there to make it stand out and look funny. 2, suggest why contraceptive pills do not contain FSH. Well, FSH is the follicle stimulating hormone. And if you remember, inside the follicle is the egg. And so the follicle stimulating hormone causes the follicle to mature, and it's obviously the egg that's inside the follicle to mature. And once that follicle and egg have matured, that follicle stimulating hormone, the FSH, stimulates the release of estrogen and LH, luteinizing hormone which causes ovulation. So basically, the FSH increases the chance of fertilization and pregnancy, which would be completely counterproductive in a pill to prevent pregnancy. So FSH stimulates a follicle to mature. FSH stimulates the release of estrogen and LH, luteinizing hormone, which would lead to ovulation. So FSH would actually increase the chances of fertilization, not something you want in a contraceptive pill. 3. Give one example of other chemical methods, which is the fourth bar, that could be included in figure 4.1. You could say the hormone implants, a hormone patch, hormone injections. You could say spermicide. You could say the IUD, intrauterine device, because that actually uses the copper on the IUD to make it so that you're unlikely to have implantation. So those are all chemical methods. I'm going to put down spermicide, but you only put down one example. You can use any of those, though. 4. State 2 methods of birth control that were not listed in the survey. Again, there's quite a few methods you could use. You could say, well, not as many as there are for chemical methods. You could say abstinence. A great method of birth control is not having intercourse. So abstinence is the most effective way of not getting pregnant. But there's other methods that can be used. You can say natural contraception or the rhythm method, which is probably the least effective way of not getting pregnant or causing pregnancy. And another way you can say is surgical sterilization. So surgical sterilization. In men, they cut the sperm ducts, vasectomies, and in women, they cut the oviducts. All right. 5. The percentage of the last 3 bars in figure 4.1 added together is 90%. Suggest why the percentage of women who used any type of contraceptive method, the first bar, is not equal to the sum of the last 3 bars. So the first bar says that about 78% of women used some form of contraceptive method. If we take 68 plus 16 plus 6, that is certainly a lot more than 78. As it says in the question, that's 90%. So why do these added together result in more than the first bar? Well, there's a couple reasons. One of them could be that some women will use more than one method. Some women may use condoms to prevent STIs and contraceptive pills to prevent pregnancy, just in case something happened, or another chemical method. So that is one highly likely method. Another possible method is that the people completing the survey didn't do it either correctly or honestly. So the highly likely answer there is that some women could have used more than one method of contraception. But as I said, you could also say that some people may not have completed the survey correctly or honestly. That also happens. That's just a problem with surveys in general. Question 5. Figure 5.1 shows an adult fly, Chrysomima megacephala. Cephala meaning head and mega meaning big, so it probably it's a big-headed fly. A. State 3 visible features from figure 5.1 that can be used to distinguish adult insects from other arthropods. So basically, what does this insect have that all adult insects should have? Obviously, larval insects don't necessarily have these parts, but adult insects should all have these parts. So one of the things all insects have is 3 pairs of legs. Okay, so 3 pairs of legs. Another thing they all have is they have 3 distinct body parts. So they have a head, a thorax, and an abdomen. Remember, many of the other arthropods only have two body parts, such as they call the cephalothorax. So the head and the thorax are combined into one pretty much. Another thing that that insects have that other arthropods don't have are antennae. They can be either be long and straight, or they can be all fluffy to catch. And quite often the fluffy types, fluffy is not the technical term, are often the males, because the fluffiness is to detect the pheromones, the hormones given off by the females. But you don't need to know that. That's just an aside. So they have a pair of antennae. And another thing you can say is that they have these big compound eyes. They're not eyes like ours, they're big compound eyes. Some of the other arthropods have much more simple eyes. B. Fly larvae are immature insects that are often used in experiments on respiration. Give the balanced chemical equation for aerobic respiration. Okay, the chemical equation basically of sugar plus oxygen produces carbon dioxide plus water. So, C6H12O6, that is glucose, that's a sugar, plus 6O2, oxygen. You obviously need oxygen for aerobic respiration. Produces 6CO2. Because we have 6 carbons in the glucose, we need 6 carbons in the carbon dioxide. Plus 6H2O, because if you count up all the other hydrogens and oxygens and the rest of the reactant side, you'll need to get more. You'll need 6H2O to balance everything else out. You could also say plus energy, but energy isn't an actual chemical. It's just a thing. But if you said plus energy, that's okay. C. A respirometer is shown in figure 5.2. It can be used to estimate an organism's rate of respiration. So this is a respirometer. Okay, we have this is a water bath to keep everything a nice consistent temperature. We have some fly larvae sitting on a wire mesh. The reason why they're on a wire mesh partway up is because some soda lime is down at the bottom. And if the fly larvae were crawling all over the soda lime, they would die because soda lime is not good for fly larvae. Okay, as they respire, they'll be producing carbon dioxide, which will be absorbed by the soda lime, and they'll be using up oxygen. So this little droplet of oil will move that way. Okay, they'll be using up the oxygen, and the carbon dioxide that they produce will be absorbed by the soda lime. All right. 1. Complete the sentences. A respirometer can be used to calculate the volume of oxygen used by the fly larva by measuring the distance the droplet of oil moves in 1 minute, or the length of the droplet of oil moves in 1 minute. A water bath is used to control the temperature of the apparatus. There we go, that's 3 marks. The soda lime in the respirometer absorbs carbon dioxide. Explain why this is important in the investigation. Well, basically, if you didn't have something absorbing the carbon dioxide, the oxygen would be used up, but the volume of gas would be replaced because carbon dioxide would be being produced. You wouldn't really be able to see any difference. So carbon dioxide will affect the volume of gas being measured. And another thing that carbon dioxide does is if you're using up all the oxygen and producing a lot of carbon dioxide will end up killing the larva. Eventually, if the experiment were to continue for way too long, the oxygen would be used up and the larva would die. Well, the droplet of water would go actually into the test tube and then oxygen could go in the tube. But if you don't remove the carbon dioxide, then carbon dioxide is heavier than oxygen, and so it would displace the oxygen, so there would be no oxygen for the larva to breathe with. 3. Fly larvae respire to release energy, just like every other organism that's not a plant. State 2 uses of energy in a fly larva. So what do they use the energy for? Oh my gosh, this is one that you should be able to get an easy 2 marks for because there are so many things you can put down. You can say it's for growth or development. You could say it's for active transport because obviously you need energy to do active transport. You need ATP. You could say it's for protein synthesis, to produce proteins. You could say it's for cell division. You could say it's for the passage of nerve impulses. You could say it's for muscle contraction. You could say it's for metabolism. You could say it's for metamorphosis, movement, breathing, egestion, digestion, excretion, anything else you could think of that uses energy. So you should be able to get 2 marks for this, but only put down 2 marks. The examiner doesn't want to mark 20 things. 2 is good enough. D. A student used a respirometer to investigate the effect of temperature on the rate of respiration of germinating seeds. Predict the results of this investigation and explain your prediction. So basically, they're looking at the rate of respiration and the effect of temperature. So our prediction should be what happens when the temperature increases. When the temperature or you can say temperature decreases. When the temperature increases, what happens? Well, as the temperature increases, the rate of respiration will, what will it do? Well, first of all, it will increase. So we have our temperature is increasing here. Our rate of respiration will increase. So as the temperature increases, the rate of respiration increases until it reaches a particular number called the optimum temperature. And I'm pretty sure you can figure out where that came from. And then it will decrease. And then decrease. Because you can't just say it will increase, because if you keep increasing the temperature, it won't just continually increase. Eventually, you'll reach the optimum temperature and the rate of respiration will start going down. Our explanation is obviously to do with enzymes. So there will be an optimum temperature for seed germination due to enzymes. So you can say, obviously, as you know, at low temperatures, there's not enough kinetic energy for effective collisions. And at high temperatures, the enzymes denature. At low temperatures, there's not enough energy for effective collisions. And at high temperatures, the enzymes denature. So that would probably be 2 marks. Okay. Another thing you can say if you wanted, but you probably have enough marks here, is that temperature will also affect the pressure of the gas in the respirometer. So as temperature increases, the pressure of the gas will increase. But that will be minimal if the respiration decreases after a certain amount of time. So that's a minimal answer. Question 6. Bacteria are useful in biotechnology and genetic engineering. Figure 6.1 shows a photomicrograph of a bacterium. Okay, this is one bacterium, but if you notice, it's kind of pinching in the middle, 2 sections of DNA. So A, state the name of the process that is taking place in figure 6.1, and that is asexual reproduction. Okay, it's doing asexual reproduction. B1, write the formula that would be used to calculate the actual width of the bacterium. So the actual width equals the image size divided by the magnification. Okay, the way that I always remember this, I always remember magnification equals the drawing or the image over actual. The image over the actual. Okay, and so then you just rearrange it and swap those 2 around. So actual equals drawing or image over the magnification. There we go. 2. The actual width of the bacterium is 0.0008 millimeters. Convert this value to micrometers. So 1 millimeter equals 1,000 micrometers. Okay, so if we want micrometers, that equals 0.0008 0.8 millimeters times 1,000 micrometers per millimeter. You can cross out the millimeters. You don't have to put in your units here. You could just say times 1,000. But if you're ever uncertain, putting the units in makes it clear which if you're doing it the right way around, if it should be multiplying or dividing. And so that equals 0.0008 times 1,000 equals 0.8 micrometers. There we go, 0.8 micrometers. C. Genetically modified bacteria can produce human insulin. 1. State the name of the disease that can be treated with insulin injections, and that is called diabetes. Okay. 2. Insulin is a protein. Describe the process of using bacteria in genetic engineering to produce human proteins. Okay, this is a 5-mark question, but it's actually quite a long question, and there's a lot of information they want in it. Because if certain parts, if you're missing it out, it's not really a very complete or correct answer. And you have to put them in the right order, so you have to know the process. So first of all, they need to isolate a human gene that codes for a human protein. So a human gene that codes for a human protein is isolated. And then they get bacteria. And in the bacteria, there's these circles of DNA called the plasmids. And those plasmids are cut using restriction enzymes. A plasmid from bacteria is cut using restriction enzymes. And using the restriction enzymes means that there are complementary sticky ends formed. So complementary sticky ends are formed. Okay, once they have the plasmid with the complementary sticky ends, they can then cut the human gene, the human DNA, with the same restriction enzyme. Human DNA is cut with the same restriction enzyme. So again, complementary sticky ends are formed. You could always obviously put the plasmid or the human DNA in either order, because it doesn't matter which one they cut first. Okay, then they put them together, and that forms recombinant DNA. So recombinant DNA is formed by putting the human gene into the plasmid. Okay, and how do they stick them together? How do they glue them together? Well, ligase, which is an enzyme, is used to join both of them together. So ligase is used to join the plasmid and the gene. I always think of ligase as the glue and the restriction enzyme as the scissors. Once the ligase is glued in the plasmid and the gene together, although don't use the word glued in, because that's not actually a word you should use. That's just how you should remember it with. The plasmid is with the gene in is then inserted back into bacteria. So the plasmid with the human gene is inserted back into bacteria. And then the bacteria with the plasmid are able to multiply and produce a protein. So the bacteria with the human gene multiply and they end up producing the human protein. And here you can put in any other valid point, such as the protein is then collected and purified. You can say that, you can mention something about identifying the transformed bacteria. So anything else that's valid to this process. This is obviously a lot more than 5 marks. There's a few things you can skip out, but still have the idea of what's happening. There's a few parts here, though, that if you miss it out, like restriction enzyme or ligase or the plasmid, it might be very difficult to get the full marks. D. Genetically modified bacteria are often grown in fermenters. 1. Suggest why steam is used to clean fermenters. Well, again, there's quite a few reasons you can use. 1 is that it kills the microorganisms, such as bacteria or fungi that shouldn't be there. So it kills microorganisms. Any microorganism that happens to be in the fermenter will then be given the ideal growing conditions to grow and reproduce, just like what they're growing in the fermenter. By killing the microorganisms, it prevents contamination. So it prevents contamination by bacteria or other microorganisms. Another thing you can say is that steam does not contaminate the product with chemicals. So if you were to sterilize a fermenter with bleach, there might be some bleach residue left behind, and that could be detrimental to the growth in the fermenter, the bacteria that are being modified in the fermenter. Okay, and another thing that steam does is if you put steam into a vessel, it will get into every single little crevice and crack and into the little bits of the pipe that are very difficult to get to. So steam reaches everywhere. So, but it's 2 marks. You should only put down two points, really. 2, state 3 conditions inside a fermenter that are measured and controlled. Oh, again, there's so many things that are measured and controlled. Basically, anything that can affect the growth of the bacteria. So one of them you can say pH. pH has to be controlled. The temperature has to be controlled. The oxygen concentration. The oxygen concentration has to be controlled. You can say the carbon dioxide concentration, the amount of nutrients that they're given, the amount of waste that's allowed to remain in the vessel, the turbidity, and turbidity just means how much it's stirred, the gas pressure, the speed of the stirring, the amount of the product produced. So there's lots of things you can put in, but these are 3 very good ones. And 3, state the name of one commercial product other than insulin that is made in fermenters. So one of them, one big, really, really, really important one is penicillin. Okay, that's a commercial product. Do not say penicillium, because penicillium is a fungus that produces penicillin. But penicillium, the fungus, is not the commercial product. The penicillin is the commercial product. Okay, so you have to be careful about your wording of this. And, I mean, there's a few other things you can put down, so any other valid point, but penicillin is the one they're really looking for. E, crop plants can also be genetically modified. Describe the advantages of genetically modifying crops. Okay, there's many advantages that you could put down. So you could say that they can result in resistance to diseases, or resistance to droughts, or they can result in resistance to extra salt in the soil, frost resistance, pest or insect resistance. Many different types of resistances can be produced from genetically modifying crops, but don't write down every one. It's only a 4-mark question. So genetically modified crops can result in resistance to insects or droughts. As I said, just choose a couple, and there's lots of things they can be resistant to. And this can produce a larger or faster yield. So this can produce larger or faster yields. Okay, another thing that you can say is that it can increase the nutritional value of certain crops. So it can increase the nutrition of certain crops. Golden rice, for example, contains a lot of extra vitamin A, so the people who eat it don't become vitamin A deficient, as there's very few other sources of vitamin A. Another thing it could be used for is vaccine production. You can actually produce vaccines through genetically modifying crops, which is very crazy, I think, but you can. So vaccine production. And, well, that's 4 points, but you could say any other valid point. There's any other valid points you could put down. You could say that by genetically modifying it, you can modify the growth. So you can make really big tomatoes. Or you could say that they can cause adaptations to harsh environments. You can grow them in places that are very difficult to grow plants. It can also result in a very fast improvement to crops, so you don't have to wait generations and generations to get the changes that you want. Through evolution alone, you can change the crops very quickly. There might be benefits to the environment for some of the genetically modified crops. Basically, in the end, most of the reasons for genetically modified crops is you get a more desirable product, and that means that the farmers get more money from the crops. And another benefit is that if there's food shortages, so benefits to humans, so if there's food shortages in areas, famines, you can produce a lot more food because you have increased yields. Okay, that's a lot of points you could put down. Reword it, but make sure you have at least 4 good points. And that is the end of this exam. I really hope you found it useful. If you have, we'd appreciate very much if you'd press the like button and if you'd subscribe if you haven't already. And we also love hearing from you. So if you have any comments or questions or anything else you'd like to say to us, please write them in the discussion section below. And have a great day.